Hi, my name is Theresa Harbig from the University of Tübingen, and today I'm presenting GoCompass, a tool for the visual navigation of multiple lists of gene ontology terms. A common experiment in biological research is to treat humans, mice, or cells with medication, such as antibiotics, and to see what happens. For example, this mouse is treated with antibiotics, which activate certain genes that are expressed in response to the treatment. These genes are associated with functions, such as cellular response to antibiotic. This term, cellular response to antibiotic, is a so-called gene ontology term, or short, a GO term. GO terms are standardized vocabulary to describe the biological function, the molecular process, or the cellular component of genes. However, we can only draw real conclusions if we do a comparison. Therefore, we always have an untreated control as a reference. In many experiments, more than one comparison is done to compare, for example, the effects of different antibiotics on mice. From these comparisons, we can then derive lists of differentially expressed genes, one list per comparison. To summarize these lists on a functional level, we often translate these lists into lists of GO terms, using a method called GO enrichment. Remember, under different conditions, different genes are expressed associated with different GO terms. For example, in mice, 14 genes are associated with cellular response to antibiotic. Of these 14 genes, 7 are differentially expressed in comparison with the untreated mice. Some of the genes are upregulated, some are downregulated. The question now is, how many genes for a GO term have to be expressed so that we include it in our final lists of GO terms? Or more specifically, are more genes differentially expressed with cellular response to antibiotic than we would expect by chance? The question is answered using a statistical overrepresentation test, which associates every GO term with a p-value, based on which we can decide if we call the GO term overrepresented or not. Eventually, we want to compare multiple of these lists, but this is not always an easy task. In a way, these lists are like shopping lists. They can contain different levels of detail, and it is hard to judge how similar they are. For example, here, Mathias' list contains vegetables and dinner, while Theresa's list is more specific. Moreover, both lists contain fruits. While Mathias' list includes oranges, Theresa's list includes tangerines, which are similar items but not the same. Similarly, dog food and cat food are both food items for animals, but how similar are they? We can construct a graph for food items to show how they are related semantically. Here in this graph, we group them based on how you might group them on your shopping list if you're a well-organized shopper. Obviously, this is a man-made construct and not a universal truth. With this graph, we can calculate a similarity value for food items, such as counting the edges between two items. When we do this for orange and tangerine, we get a value of 2. While when we compare cucumber and orange, we get a value of 4. Therefore, according to this graph, we can deduce that oranges and tangerines are more closely related than oranges and cucumbers. We call this calculated value the semantic similarity between two items. However, there are more advanced methods in practice that combat some of the issues with graphs like this. For example, the graph might be unbalanced. In fact, for this graph, one could argue that oranges and tangerines should be more closely related than oranges and apples because they're both citrus fruits. GO terms are organized in a similar way using a graph structure, which is defined by the Gene Ontology consortium. This means that GO terms are in parent-child relationships. Our term, cellular response to antibiotic, has response to antibiotic and cellular response to chemical as parents. Therefore, if a gene is associated with a specific GO term, it is also associated with all its parents. So if we find specific GO terms in our list, we might find all their parents as well. However, often the parents are dispensable, since they do not add much information and increase the length of the list. This brings us to the actual tool, GoCompass. We had three main goals for the development of GoCompass: reducing the dispensability and therefore the length of the lists with the help of the underlying GO graph, comparing lists effectively in a visualization, and also incorporating gene information in the visualization. GoCompass can take as input either multiple lists of genes that it translates into GO terms using GO enrichment analysis, or it can use GO terms directly. For our first goal, reducing the dispensability, we cluster the GO terms hierarchically. Moreover, we allow users to define their own dispensability cutoff. It is important to say here that the resulting hierarchy is a new hierarchical clustering and not the original Go graph. For the dispensability clustering, the union of the GO terms is taken, and for every pair of terms, semantic similarity is calculated using a method chosen by the user. The pairs are then sorted by their similarity. For each pair, we reject one of the terms based on several criteria. When a term is rejected, it is placed as the child of the accepted term in the pair in the hierarchical clustering. In general, less specific terms and less significant terms are rejected in favor of specific significant terms. The first criteria is term frequency. A term that is associated with more than 5% of all genes in the annotation is rejected in favor of the rarer term. The second criteria is significance. A term that is less significant by a specific margin than in the majority of the lists is rejected. Finally, the relationship of the terms in the original graph is taken into account. If the two compared terms are in a parent-child relationship, the child is rejected unless it is associated with most of the same genes as its parent. This results in a hierarchy where dispensable terms are placed further away from the root than non-dispensable terms. This hierarchy is the core visualization of GoCompass. On the right side of the hierarchy, a heat map is aligned, showing the significance of the GO terms for each of the lists. The darker the shade of red, the more significant. If a cell is marked with a dot, it means that the p-value is below a significance threshold defined by the user, for example, below 0.05. In this case, we compare 5 lists. For this experiment, a mouse has been treated with an antibiotic, and samples have been taken at 5 time points. These time points have been compared with each other to find differentially expressed genes, for example, day 8 versus day 0. This results in 6 pairwise comparisons, of which 5 showed significantly differentially expressed genes. These are the ones included in this analysis. 2 sliders in this visualization help preparing the data for the other comparative visualizations. With the right slider, GO terms can be filtered out interactively, allowing users to decide on the desired level of redundancy in the lists. With the left slider, the tree is cut to produce flat clusters of terms, according to what users perceive as semantically similar. With the cluster slider set to 0.2, we get 7 well-defined clusters. For example, the dark green cluster contains GO terms related to blood vessel development. The lilac cluster is related to cell localization, and the yellow cluster is related to regulation. In addition to this component, for comparing lists, the GoCompass dashboard has 3 other main components, each with a different focus. Moving the filtering and clustering sliders has an effect on all other visualizations. Next, we are going to see how similar the lists are on a global level. This heat map shows the correlation of p-values between the different lists. Thereby, users can gain a quick overview which lists are similar and which are distinct. Here, one list, day 8 versus day 0, seems to be very dissimilar to the other lists, indicated by the negative correlation. However, there is one list, day 18 versus day 8, with which it has a slightly positive correlation. We now want to compare these two lists in more detail. We can do this using an adjacent bar chart visualization. This view shows a bar chart for each selected list with bars corresponding to geoterms. The height of the bar indicates the negative logarithm of the p-value. Therefore, the higher, the more significant. The dotted line shows our current significant threshold. We find that the two lists share two significant geoterms: rhythmic process and regulation of circadian rhythm. We now look at exactly these terms in the tree map visualization and see how the underlying genes are expressed. The tree map visualizes the composition of a single list where the area of the rectangle corresponds to the significance of the geoterms. In the tree map rectangle, we placed a small glyph visualization that encodes gene information. Since geoterms are associated with sets of genes, there is 3 values that we need to visualize: the set size, the number of genes that are expressed, and their expression values. The color of the bar of the glyph shows the median gene expression, which is negative in this case. The small attachment on top ensures that the color is always visible, even when the bar is very small. The length of the bar shows the proportion of genes that are differentially expressed. Here it's about a quarter of the genes. The background of the glyph encodes for the set size, which is the total number of genes associated with the geoterm, while smaller sets receive brighter colors than larger sets. Transitions between tree maps are animated to allow users to see the most prominent changes. We can now see how the genes associated with the geoterms are expressed under different conditions. While the expression is lower in day 18 than in day 8 as indicated by the blue color, it is higher in day 8 than in day 0, which means that it's actually highest at day 8. For a more detailed gene-centric analysis, the data can be exported as a simple CSV file. I've now given you a sneak peek for all 4 main components of GoCompass. Using the clustering visualization, users can interactively filter out dispensable geoterms. The tree map is a list-centric view that incorporates gene information. With the correlation heat map, lists are compared using a single value. And finally, in the detailed comparison, a subset of lists can be compared using bar charts. In summary, GoCompass simultaneously reduces the length of the lists and their redundancy. It provides a comparative visualization and incorporates gene information. With the visualized dispensability clustering, it increases the explainability of the underlying algorithm. The gene lists that it uses as input can come from many different sources. Differential expression is just one example. It can also use multi-omics data as input or data from multi-species experiments. Now I want to thank you for your attention, and here with this QR code you can find a link to our application on our TUVIS visualization server. Also, I want to thank the authors of the package Goa tools who implemented a specialized semantic similarity method for us, and also our experts for the expert feedback sessions, especially Dr. Angus Lavelle, who also helped us to read in the data of his study for our case study.